So today's lecture is on RNA-seq. So this RNA-seq, that is RNA sequencing, was first developed in mid 2000 with the advent of next generation sequencing. So RNA-seq, that is named an abbreviation of RNA sequencing. So it is a particular technology-based sequencing technique which uses next generation sequencing to reveal the presence and quantify RNA in a biological sample at a given moment, analyzing the continuously changing cellular transcriptome. So what RNA is? RNA is an abbreviation which given for RNA sequence, and this RNA is a technology-based sequencing technique. So this RNA use of next generation sequencing techniques. So next generation sequencing techniques are the sequence of RNA molecule, so that it will reveal the presence and quantity of RNA in one moment. Suppose at particular moment we are taking a biological sample and we are analyzing quantity RNA particular for that moment, and also analyzing continuously changing cellular system. So first we will take the biological sample for a moment, then after certain changes we will take again that particular biological sample, and again we will analyze the changes that take place in the transcriptome. Specifically, RNA-seq facilitates the ability to look at alternative gene splice transcripts, post-transcriptional modifications, gene fusion, mutations, or single nucleotide polymorphisms, and changes in gene expression over time, or differences in gene expression in different groups or treatments. So specifically this RNA-seq, it will facilitate the ability so that one can look at alternative gene splice transcripts. So where splicing has been taken place in the transcript, post-transcriptional modification we can also analyze from RNA-seq. Then gene fusion is there, mutation or any of the nucleotide polymorphism we can understand, and changes in the gene expression over the time, or differences in the gene expression in different groups. For example, taking control cell and we are taking the cancerous cell. So we can find out the difference in the gene expression pattern of those different cells. In addition to mRNA transcripts, RNA-seq can look at different populations of RNA to include total RNA, small RNA, such as miRNA, tRNA, and ribosomal profiling. So in this RNA-seq, we are sequencing the mRNA molecule. So in addition to this mRNA molecule, we can also take the different populations of the RNA, such as tRNA is there, then small RNA is there, then miRNA, ribosomal profiling. So these we can also analyze by using RNA-seq. Then RNA-seq can also be used to determine exon or intron boundaries and verify or amend previously annotated 5' and 3' gene boundaries. So recent advances in RNA-seq include single cell sequencing and in situ sequencing of fixed tissue. So this RNA-seq can be used to find out the boundaries of introns of exons, So where that particular boundary of intron is there, or where the exon ends. So this we can find out or determine by using the RNA-seq technique. Then by using this technique, we can also verify or previously annotated 5' and 3' gene boundaries. So whatever previously annotation is done regarding the 5' or 3' boundaries, so this also we can verify by using the RNA technique. And recent advances in RNA-seq include single cell sequencing and in situ fixed tissue. So we can also do in situ sequencing of a fixed tissue, or can also do single cell sequencing. So these are the advances in RNA-seq technique. So before going to the principle of RNA-seq, we will see why to sequence the RNA versus DNA. So because sequencing, because it is involved in the functional studies. So in the last lecture, I have told that genome is constant, or DNA composition in all the cell types of a particular organism, it's same, but an has a pronounced effect on gene expression. So that particular experimental condition that have a different effect on that particular gene expression. For example, if we take the example of a cell line, so we will take just a cell line, and we will take another cell line to which we are going to find out the effect of it. So the experimental condition is different in both the types of, but if we took if we look at the genome of both the cell line, the genome may be same, but the experimental condition is same. In the first cell line, when any of the drug, and in the drug. So the experimental condition that are having a different effect on the gene expression. Then next will be the predicting the transcript sequence from the genome sequence is very difficult. So if we have completed the genome sequence of a particular organism, and based on that genome sequence to find out which is the transcript sequence, it is very difficult. And some of the molecular features can only observed at the RNA level. So such features cannot be observed at the DNA level. So such examples of such features are alternative isoforms, then fusion transcripts, RNA editing. So that's why we have to sequence the RNA molecule. So next we will see the principle of RNA sequencing. So RNA sequencing is a next generation high throughput RNA sequencing and quantification method that is used for studying the transcriptomics and gene expression. So this RNA sequencing, it is based on the next generation sequencing methods, and it gives a high throughput RNA sequencing. A large data will be generated, and we can also find out the gene expression by using the RNA sequencing. And so that's why it is used for the studying of transcriptomics. So many transcriptomes we can study by using the RNA-seq technique. So the principle of this RNA-seq is a cDNA, it's constructed from total mRNA through the process of reverse transcription and fragmented. Then simultaneously, adapter ligation and library preparation are practiced before doing sequencing. Then the sequencer reads and quantifies the cDNA complementary to the mRNA. So the principle of this RNA-seq is that we have to construct the cDNA from the total mRNA. So for this, what we will be required? First, we have to isolate the RNA molecule. Then we have to purify the mRNA molecule from the total RNA. Then after reverse transcription, we have to produce the cDNA molecule. So after producing the cDNA molecule, we have to fragment this cDNA molecule. And afterwards, a adapter ligation and library preparation are practiced. So if we see the features of the next generation sequencing techniques, so there are steps like library preparation, then adapter ligation. So this RNA-seq is based on the next generation sequencing, right? So that's why we have to carry out this procedure, that is adapter ligation, then library preparation. So after this, we have to carry out the sequencing. Then the sequencer reads and quantifies the cDNA, which is complementary to the mRNA. So after analysis of that particular data, we can get to know the cDNA, which is complementary to the mRNA, and we have to quantify this. And afterwards, we can find out the gene expression or differential gene expression. The procedure in the RNA-seq, first is RNA isolation, second is cDNA synthesis, third step is adapter ligation, fourth step is library preparation. Then afterwards, DNA has to be fragmented. Then it is then sequenced and the data has to be analyzed. So this is the workflow of RNA-seq technique. So in vivo, this is a particular genome. So from the process of synthesizing RNA from the DNA, it is known as the transcription. So by using this information, RNA will be synthesized. So this is the pre-RNA or precursor mRNA. So precursor mRNA contains introns and exons. So these are the exons and this is the intron. So two introns are there. So after intron splicing, that means after removing this intron, mRNA get produced. So this is this. So what we have to do, we have to isolate the mRNA. After that, we have to fragment these mRNA molecules. So for this, we can use the restriction enzymes. So for this, we can use the enzymes so that RNA can get fragmented. Then afterwards, we have to carry out the reverse transcription so that complementary DNA molecule to this corresponding mRNA molecule will get produced. So afterwards, double-stranded cDNA molecules will be formed. Then after this, we have to sequence these fragments. These double-stranded cDNA fragments has to be sequenced by using next generation sequencing method. For example, we can take the method like Illumina sequencing. So after that, we can get to know the sequence of each double-stranded cDNA fragment. After that, the next is in silico method. That means we have to use the computer and we have to analyze this data by using different softwares. So we have to process this sequence. So we have to align these sequences and then afterwards we have to find out the variants in the gene expression. So this particular is a splice variant A, and this is splice variant B. First step is RNA isolation. The first step in the RNA-seq is isolation of total RNA, mRNA, or non-coding RNA for the experiment. Then after isolating the RNA, this isolation of RNA is a tedious process in comparison to the DNA isolation. The RNA can be break down easily. So also there are chances of contamination for the RNA is very high. So RNA isolation is a very tedious job as compared to the DNA isolation. Because RNA molecule is not stable, DNA molecule is much stable. And there are also the chances of contamination, that RNA may get contaminated. So I have already told you that, for example, if we are going to isolate the RNA in the laboratory, we have to use the gloves, because in our hands there are many RNAs are present. So that will degrade the RNA molecule. So we have to take extra precaution for isolating the RNA molecule, so that that particular RNA will not get contaminated. High yield pure RNA is must required for RNA sequencing. So the yield of RNA molecule should be high and it should be extra pure. This is the pre-requirement for the RNA sequencing. Then the purity and quantity of the RNA are measured using nanodrop. Then next, after getting the RNA molecule, mRNA molecule, we have to carry out the reverse transcription. So reverse transcriptase PCR we use for the reverse transcription process. So another innovation setup for RNA sequencing is to do reverse transcriptase PCR, in which RNA is reverse transcribed into DNA. So using a special type of PCR, using a special type of polymerase that is known as DNA versus transcriptase, the cDNA is synthesized from the mRNA molecule. So by using this, we can do synthesis of cDNA molecule from the mRNA molecule. So for this special type of polymerase enzyme is used, that is DNA reverse transcriptase. Then next, second strand cDNA synthesis. So after the cDNA synthesized from the mRNA, the second strand DNA synthesis is must required, because we require double stranded cDNA molecule. So for that, PCR is performed using the normal tag DNA polymerase in the conventional PCR, so that the complementary strand to that particular cDNA molecule will get formed and we can get the double stranded cDNA molecule. Then polymerase adds DNTP to the growing DNA strand using the primer set, and we can get the double stranded cDNA molecule. So we, by using that particular reverse transcription, we can get many fragments of cDNA molecules, because earlier we have carried out the digestion of that particular RNA molecule. So next step is the library preparation. The first step in the library preparation or next generation library preparation starts with the fragmentation. So using the special type of restriction endonucleases, whole sets of double-stranded cDNA is fragmented for the next generation sequencing. That means we have to do a restriction digestion so that we can get different double-stranded cDNA molecules. And from that, we can construct the library. The library preparation varies from platform to platform, as some fragments mRNA before performing reverse transcriptase, while some perform it later after preparation of cDNA. So that step I told you that we have to carry out the fragmentation of this RNA molecule. Otherwise, we have to carry out the reverse transcription so that we can get the double-stranded cDNA molecule. And after getting the double-stranded cDNA molecule, we have to fragment this double-stranded cDNA molecule. So ultimately, we can get the small fragments of double-stranded cDNA molecule. So it depends on the platform which we are going to use in the sequencing process. So in the last step of the library preparation, the ends are ligated with the adapter sequencers and amplified for repairing the ends. So for the library preparation, whatever the ends of that particular cDNA fragments will be there, that we have to ligate with the adapter sequence. And by using those adapter sequence, we have to amplify those particular cDNA fragments. So for this, we can also do a repairing step. So DA tailing is optional in case of mRNA library. So after preparation of library, we have to purify the library. That is library purification. So the entire library is purified using the ready-to-use DNA purification kits. So many commercially available kits are present. So we can use those kits, and by using those kits, we can purify the entire library. So in the last step, it is very crucial to purify the entire fragment library. For that, the concentration of the library is assessed using the quantitative PCR or bioanalyzer. So we have to purify that library, and after that we have to concentrate that particular library. It means we have to quantify that particular library. So for this, quantitative PCR can be used, or we can also use the bioanalyzer. The next step is DNA sequencing. That means that particular complementary DNA sequencing. So now the sample is sequenced in the high throughput next generation sequencing machine, which reads the sequence as well as quantifies the nucleic acid as well. So we have to carry out the sequencing step. So the chemistry behind the sequencing the cDNA is depend on platform that we are using. Although widely used method is used for fluorescence chemistry. So depending on the platform, we have modifiers and we have out the sequencing. So in the sequencer, individual fragments are read separately and sequenced. The final data are sent to the bioinformatics lab for post-sequencing analysis. So here we can use Illumina sequencing or ion torrent sequencing, or we can also use the ABI solid sequencing. So any of the method we can use, but depending on the method, some of the protocol. So after getting that particular result, analyze that particular data by using certain bioinformatics tools. Transcriptome data analysis. So different fragments are sequenced in the machine and data are collected. In the next step, based on the flanking region of the fragments, the so-called contigs are arranged for getting the splice variants. So in the data analysis, we have to find out the flanking regions of those particular fragments. So whenever where there is a flanking region, that portion we can use for that particular contigs. That means we can continuously arrange those fragments so that we can get the splice variants. In the next step, the transcriptome is compared with the reference genome, reference sequence, or can be assembled de novo. So next step will be we have to compare this transcriptome with the reference sequence. So this I told regarding the mRNA molecule and sequencing of that particular mRNA. So there may be a small RNA or non-coding RNA in that particular cell. So that also we can sequence by using the RNA-seq technique. So when sequencing the RNA other than mRNA, the library preparation is modified. So the cellular RNA is selected based on the desired size range. So for small RNA targets such as miRNA, the RNA is isolated through site selection. So this can be performed with a site exclusion gel through site selection magnetic beads or with a commercially developed kit. So once isolated, the linkers are added to the 3' and 5' end and then purified. Then the final step will be cDNA generation through the reverse transcription. So if we have to sequence the small RNA or non-coding RNA, we have to carry out some of the modification. So the modification will be, we have to select that particular RNA based on the desired size range. So we just take the example of miRNA. So for this, RNA has to be isolated through the site selection. So for this, size exclusion gel can be used and magnetic beads are used. So through this, we can find out the that particular RNA of a desired size. And after getting that, we have to add that particular linker, and after this we have to carry out the reverse transcription. So the next is we can also use that particular RNA molecule, and we have to we can sequence that particular RNA molecule. So direct RNA sequencing. So because converting the RNA into cDNA, then ligation, amplification, and other sample manipulations have been shown to introduce biases and artifacts that may interfere with both the proper characterization and quantification of the transcripts. Single molecule direct RNA sequencing has been explored by companies including Helicos, Oxford Nanopore Technologies, and others. So this technology sequences RNA molecules directly in a massively parallel manner. So we can directly take the RNA molecule and we can sequence that RNA molecule apart from converting that particular RNA into the cDNA. So after getting the cDNA, we have to prepare the library. So for this, we have to add the linkers, the adapters, and then we have to amplify this, and after this we have to sequence. So by skipping this step, why we have to skip this step? Because that steps may introduce biases and it artifacts that it may interfere with both proper characterization and quantification of transcript. So whatever the result we can get regarding the characterization and quantification of transcript, that may get vary. So to avoid this, we can directly use the RNA molecule for the direct sequencing. So for this, we have already studied this Oxford Nanopore technology. So different techniques are available. Different companies provide different machines, and by using that machines, we can directly get the sequence of that particular RNA molecule. So this is about the RNA-seq technique.